interesting because this slide describes the heart of our chemistry, and this part of our technology is actually generic and is not application specific. So regardless of whether you're using our technology for microbiome sequencing or transcriptome sequencing or just general amplicon sequencing, this part of our technology is actually generic and it applies to all types of loop genomics single molecule long read sequencing. So let me go through this and show you how we're able to accomplish long read sequencing on a short read sequencer. So the first part of every loop genomics prep, and this is kind of just a summary of the previous slide, is attaching the loop adapter to the single mRNA molecule. In the second part of the prep, each full-length mRNA molecule is amplified. So we're making multiple copies of each mRNA molecule. So now each mRNA molecule that has a unique sample index and a unique molecular identifier is now present in the sample in multiple copies. And just to remind you, at this stage, all the samples are already multiplexed into a single tube. So this reaction here, where we amplify the cDNA, in this reaction, we already have 8 or 24 or 96 samples multiplexed into a single tube, and we're just 45 minutes into the prep. Okay, now once we have every molecule amplified, we progress to the final part of the prep, and this is what we call our distribution system. In this system, and this is really the heart of our chemistry, we've developed a series of enzymes, kind of an enzymatic system that is essentially a copy-paste operation, an intramolecular copy-paste operation in DNA. So what these enzymes do is they take this loop adapter that contains the UMI and the sample index, and they copy and paste them into multiple positions within the same molecule, but not to neighboring molecules. So again, this loop adapter in its entirety, both the sample index and the molecular identifier, are copied and pasted randomly into multiple positions within the same molecule, but not to neighboring molecules. So, and obviously this is done in parallel over millions of molecules in the same tube. All right, so at this point, as you might imagine, we've already solved the hardest problem, which is getting the UMI spread out into multiple positions within the same molecule. Now, after we've done this, let me jump to the next slide and show you how we're using this to do long read sequencing. So again, these are the long molecules that were amplified with PCR. We've copied and pasted the barcodes into multiple positions within the same molecule. And then and after we've done that, we can now take this library of molecules and sequence it and basically chop it down and sequence it using regular short read sequencing. So after we do the intramolecular barcode distribution, we then go on and prepare the library for sequencing using the regular short read sequencing workflow, where molecules are just chopped down to 200 or 300 base pairs, the read length of Illumina sequencing. And what we generate are these reads that you can see here, these short reads. In the library that we sequenced, every that we load on the sequencer, every short read always starts from the loop adapter and then followed by the actual sequence of the molecule. So what our software then does, it takes the reads off of the sequencer and it clusters them by their sample index and then by their molecular identifier. And what we do is we take all the short reads that share the same barcode, the same unique molecular identifier, and we use them in order to reconstruct the sequence of the original long RNA molecule that was tagged by that same barcode. I'll repeat that. So we take all the short reads that share the same UMI, the same barcode, and we use their software in order to de novo reconstruct computationally the sequence of the original long RNA molecule that was tagged by that UMI. And that, in a nutshell, is how we're able to accomplish long read sequencing using a short read sequencer. And this ability stems from this enzymatic reaction, from our ability to copy and paste the barcode intramolecularly into multiple positions. That means that every part of the long molecule will be tagged by the same UMI because it's intramolecular. And then when we make short reads out of those molecules, every short read that originated from the same molecule will have the same UMI, which means that if we group all the short reads that share the same UMI, we'll be able to reconstruct the sequence of the original long molecule